Hi, my name is Angelica Owen, and this is my individual project 2 on DNA. History of DNA. Biologists in the 1940s had difficulty in accepting DNA as the genetic material because of the simplicity of its chemistry. It was known to be a long polymer composed of only 4 types of subunits, which resemble one another chemically. In the early 1950s, DNA was first examined by X-ray diffraction analysis, a technique for determining the three-dimensional atomic structure of a molecule. And here is a picture of an X-ray of DNA. What is DNA? DNA, or deoxyribonucleic acid, is a long molecule that contains our unique genetic code. Think of it as a recipe book. It holds all the instructions for making all the proteins in our bodies. DNA consists of 4 basic building blocks: adenine, cytosine, guanine, and thymine. The order of these bases form the instructions in the genome. DNA is a 2-stranded molecule and has a unique double helix shape like a twisted ladder. And here's a picture of DNA and the 4 nitrogenous bases. Where is it found? In eukaryotic organisms, DNA is found inside a special area of the cell called the nucleus. Because the cell is very small and organisms have many DNA molecules per cell, each DNA molecule must be tightly packaged. A packaged form of DNA is called a chromosome. And here's a picture of a eukaryotic cell with diagrams showing the in-depth parts of it all the way to the DNA. DNA replication. DNA replication, the process by which DNA makes a copy of itself during cell division. It begins when an enzyme breaks the bonds between complementary bases in DNA. This exposes the bases inside the molecules so they can be read by another enzyme and used to build 2 new DNA strands with complementary bases. The 2 daughter molecules that result each contain 1 strand from the parent molecule and 1 new strand that is complementary to it. As a result, the 2 daughter molecules are both identical to the parent molecule. DNA replication is a semi-conservative process because half of the parent DNA molecule is conserved in each of the 2 daughter DNA molecules. DNA replication. Here is a diagram of that process. DNA mutation. Mutation, which may arise during replication and/or recombination, is a permanent change in the nucleotide sequence of DNA. Damaged DNA can be mutated either by substitutions, deletions, or insertion of base pairs. For the most part, mutations are harmless, except when they lead to cell death or tumor formation. Base substitutions. Single base substitutions are called point mutations. Point mutations are the most common type of mutations. There are 2 types. Transition occurs when a purine is substituted with another purine, or when a pyrimidine is substituted with another pyrimidine. And transversion, when a purine is substituted for a pyrimidine, or a pyrimidine replaces a purine. And here's a picture of transition and transversions. Point mutations. Point mutations that occur in DNA sequencing are either silent, missense, or nonsense. A silent mutation is a base substitution occurs in 3rd position of the codon. There's a good chance that a synonymous codon will be generated. Thus, the amino acid sequence encoded by the gene is not changed, and the mutation is said to be silent. Here's an original sequence versus a point mutation. Continued. Missense, when a base substitution results in the generation of a codon that specifies a different amino acid and hence leads to a different polypeptide sequence. Depending on the type of amino acid substitution, the missense mutation is either conservative or non-conservative. Nonsense, when a base substitution results in a stop codon, ultimately truncating translation and most likely leading to a non-functioning protein. Deletions. A deletion resulting in a frame shift results when one or more base pairs are lost from the DNA. If one or more bases are deleted, the translational frame is altered, resulting in a garbled message and a non-functional product. A deletion of 3 or more bases leave the reading frame intact, and a deletion of 1 or more codons result in a protein missing or more amino acids. Insertions. The insertion of additional base pairs may lead to frame shifts depending on whether or not multiples of 3 base pairs are inserted. Combinations of insertions and deletions leading to a variety of outcomes are also possible. And here is a picture of all of the different types of mutations. Silent, nonsense, missense, a frame shift, insertion and deletion, and an insertion. Cause of mutations. Errors in DNA replication. They're actually very rare, and DNA polymerase will incorporate a non-complementary base in the daughter strand. Errors in DNA recombination. DNA often rearranges itself by a process called recombination, which proceeds via a variety of mechanisms. Occasionally, DNA is lost during replication, leading to a mutation. Chemical damage to DNA. Many chemical mutagens, some exogenous, some man-made, some environmental, are capable of damaging DNA. Radiation. Gamma rays, X-rays, even UV light can interact with compounds in the cell generating free radicals, which cause chemical damage to DNA. DNA repair. Damaged DNA can be repaired by several different mechanisms. A mismatch repair, which relies on a secondary signal with DNA to distinguish between the parental strand and the daughter strand. Nucleotide excision repair removes damaged DNA induced by ultraviolet light. Direct repair of damaged DNA. Sometimes damage to a base can be directly repaired by specialized enzymes without having to excise the nucleotide. And recombination repair. This mechanism enables a cell to replicate past the damage and fix it later. Progeria. Hutchinson-Gilford progeria syndrome, progeria or HGPS, is a rare fatal genetic condition characterized by the appearance of accelerated aging in children. Its name is derived from the Greek and means prematurely old. There are different forms of progeria, but the classical type is Hutchinson-Gilford progeria syndrome, which was named after the doctors who first described it in England. In 1886 by Dr. Jonathan Hutchinson and in 1879 by Dr. Hastings Gilford. And here's a picture of a progeria mutation. The gene LMNA. HGPS is caused by a mutation in the gene called lamina. The lamina gene produced in the lamin A protein, which is the structural scaffolding that holds the nucleus of a cell together. Researchers now believe that the defective lamin A protein makes the nucleus unstable. That cellular instability appears to lead to the process of premature aging and progeria. The lamina gene is an essential scaffolding supporting component in the nuclear envelope, which is the membrane that surrounds the nucleus. The altered lamina gene makes the nuclear envelope unstable and progressively damages the nucleus, making cells more likely to die prematurely. And here is a picture of the lamina gene. Lamina gene location. The lamina gene is located on the long Q arm of chromosome 1 at position 22. More precisely, the lamina gene is located from base pair 1,056,000. Oh my gosh, I can't even think right now. I'm sorry. Here's a lamina gene location. Characteristics of progeria. Although they're born looking healthy, children with progeria begin to display many characteristics of accelerated aging within the first 2 years of life. Signs include growth failure, loss of body fat and hair, age-looking skin, stiffness of joints, hip dislocation, generalized atherosclerosis, cardiovascular disease, and stroke. The children have a remarkably similar appearance despite differing ethnic backgrounds. Most children with progeria die of atherosclerosis at an average age of 14 years. Progeria almost always results from a new mutation rather than being inherited from a parent. Management focuses on the individual signs and symptoms of the condition, and there is currently no cure, but research involving treatment is ongoing and progress is being made. Here is a picture of a diagram of a child suffering from progeria, and here are my references.